A very delightful morning to all my loving students. This is Alpna Shukla, and once again I welcome back to you all in biology class. As we are learning about the molecular basis of inheritance, in our previous lecture we have learned about the structure of DNA, and today we are going to learn about the central dogma. So one-way flow of genetic information from DNA to protein is called central dogma. Means the information is passing from the DNA to RNA and from RNA to protein. Packing of DNA in prokaryotics. Prokaryotic do not have definite nucleus. The DNA is not secreted throughout the cell. It is held together with some protein in a region is called nucleide. The DNA in nucleide is organized in large loop held by proteins. Packing of DNA in eukaryotic. In eukaryotic, DNA is stabilized with the positively charged basic protein called the histone. Histones are the positively charged due to the rich in basic amino acid like lysine and the arginine. Histones are the organized to form a unit of 8 molecules called the histone octamer. A negatively charged DNA wrapped around the positively charged histone octamer to form a structure called the nucleosome. The nucleosome are seen as the bed on string. Nucleosome from the repeating unit of a structure is nucleus called the chromatin. And the chromatin is the packed to form a chromatin fiber. These are further coiled and condensed at the metaphase stage to form chromosome. Euchromatin in the nucleus, some loosely coiled region of chromatin is called the euchromatin. Heterochromatin in the nucleus, more dense packed region of chromatin are called heterochromatin. Euchromatin is the transferably active than the heterochromatin. Transforming principle. Faddeus Graef conduct experiment to show the transforming principle in bacteria. He conducted an experiment on mice and pneumonia bacteria, Staphylococcus pneumonias. These bacteria are found in two strains as the virulent smooth strain, non-virulent rough strain. He shows that how information passing from one generation to another generation or from one DNA to another DNA. The strain bacteria produce capsule and it's pathogenic, and the R strain lack capsule and is non-pathogenic. When the R strain are injected into the mouse, it is non-pathogenic and does not cause pneumonia. The mouse continued to live. When the S strain are injected into the mouse, that cause the pneumonia and mouse die. When heat killed S strain and inject into the mouse, that does not cause pneumonia, the mouse continued to live. When the heat kill S strain and R strain are mixed and inject into the mouse, they cause the pneumonia and mouse die. So what was the conclusion of this experiment? The conclusion was R strain bacteria had been the transferred by the heat killed S strain bacteria, and the transformation of R strain to S strain is due to transfer of genetic material. The biochemical nature of genetic material was not defined from his experiment. So that's all for today. In next class we will learn more about the genetic material. Thank you and have a nice day.